This is a demonstration on how to use the single cell expression visualizer, or SCexV. So from this page we can upload our expression data and our index sourcing data as well. So we click on browse and we can select all of our expression data, and we can do the same with our index sourcing data as well. And the points and notes here is that the files are linked on the keyword array 1, 2 and 3. So the program will automatically pair the right expression data with the right index sourcing data. So when the files have loaded, you'll be presented with some more options down the side here. And here we can choose, for example, a positive control gene, so for example beta-actin, and we can also choose GAP-DH as well. So you'll see histograms appearing for each of these, and the green bars are the CT values across all 3 plates. The lines are the density plots for individual plates. So what you hope to see are the lines on top of each other. If one was a significant outlier, then you would think that plate would require extra attention and maybe to go back and QC it again. So then we have the option to remove any cell if more than one of these has a CT value greater than 25. So we can apply this, and any cell fulfilling that criteria will then be removed. So the histograms will then change accordingly, and we can then check those. And when we're happy with those, we can just click Done here and then go through to the analysis page. So the first thing that you'll see when it comes up is a violin plot and also the principal component analysis as well. And this is what they look like. So you can see here that the PCA plot is in 3 dimensions and is fully interactive, so we've implemented this using WebGL. And you can see here that we have 2 broad groups, and the balls are coloured by the plate ID. So you can see that the groups are made up by equal representation of each of the plates, and this is what we expect to see because each of the plates was sorted using the same sorting strategy. On the left hand side you'll see violin plots for particular genes. So for example we can click on GATA1 for example, and we can see here generally that across all 3 plates its expression is by and large the same, and this is what we expect to see. If we scroll further down, you will see heat maps for the expression data, and again it's color coded by the plate they came from, and it looks pretty random. It's not been clustered, and again this is what we expect to see, and the same also applies for the index sorting underneath. We can then take a look at a dendrogram of the expression data, and here things start to clarify a lot more. You can see, for example, that we have a cluster here which is high in the erythroid genes like GATA1, EPO receptor and GFI1B. So to actually cluster the data, we can go back up to the top here, and we want to cluster on the data values, not on how they cluster in space here. I want to cluster on the expression data, and let's just say, for example, we think there are 5 clusters in there. So we can then run the analysis, and it will then cluster the data as I said, and then repopulate the page with the updated plots. Okay, so now that's done, you can see here that the spheres have been recolored, and you can see there are quite clearly some clusters here worth looking at, and also the violin plots on the left have been updated as well. So regarding that erythroid cluster, we can then take a look at GATA1 again. And here we can see quite clearly that expression is quite high in the blue cluster, it goes down a bit in the green cluster, lower still in yellow, and it's basically not present in the purple. If we scroll down a bit further, we can then look at the updated heat map. So now they're coloured by cluster, and the same also goes for the surface marker expression as well. So regarding our erythroid cluster here, you can see that the surface markers involved are CD105 and CD55, but exclusive of CD115. So you can see that it's a very quick way of getting an overview of your data, clustering it, and then finding which surface markers are responsible for the separation that you're seeing in your plots. So to say further, you can also do 2 component plots as well, and where groups tend to merge together and the separation is less clear, we've also implemented PCA plots but using densities instead, where you can see the space they occupy too. But for our purposes, the scatter plot is absolutely sufficient.